So anyway, yeah, pretty cool microbiology stuff going on. Anytime your day involves going to some place called the door to hell, um, is again like pretty badass in my opinion. All right, so again, today what we're going to be talking about are the pathogens that are not gram positive or gram negative. Okay, these are going to be things like the spirochetes, the curviform bacteria, um, and the mycoplasmas. We talked about mycoplasma a little bit when we were talking about bacterial structures and how they are mostly pleomorphic because they have a sterol composed membrane, which is much less rigid. And so they don't have these more typical rod or coccus shaped morphology. And so these are some of the things, these are some of the organisms that fall into these miscellaneous categories. We have syphilis, leptospira, and the Borrelia, which is the causative agent of Lyme disease, the Vibrio cholera, and Campylobacter, which is going to include H. pylori. Okay. And then this table goes into kind of some of the areas that they infect. And so with, for instance, syphilis, it starts out as a skin infection and then moves from there into, you know, the nervous system, the musculature, and can eventually infect the brain as well as cardiovascular tissue. Okay. We also have rickettsia. We have such as like Rocky Mountain spotted fever, and I don't know if you've heard of many of these other ones, but chlamydia, and then of course mycoplasma. Okay. So these are the outliers in the realm of bacterial pathogens, but nonetheless have a pretty important place in pathogenesis. The spirochetes, if you'll remember, are not rods or cocci. They are these spiral shaped or helical shaped bacterial cells. They can be present in the mouth, in the intestine, and often in the perigenital regions, okay, because they can be sexually transmitted diseases. They are strict parasites typically, meaning that they have to have a host, they're obligate. And in order to cultivate them in the lab, you have to have live cells oftentimes. So these are not bacteria that lend themselves to growing on a plate of agar, all right? Because they are strict parasites, because they are obligate, they need to have some kind of host cell in order to grow and divide. Remember also that these spirochetes often have that flagella that's inside the membrane, not on the outside of the cell, which helps by creating like a torsional effect for locomotion.
Well, sorry, can I ask a quick question?
Yeah, of course, please.
Would you be able to grow those in a plate with like HeLa cells, since those are immortal?
Yeah. It depends really on the particular cell type that these different pathogens will infect. So HeLa cells are cervical cells. A lot of these cells, or a lot of these pathogens, like I said, will, depending on which type, will infect epithelial cells, nervous system cells, muscular cells. So I don't know if HeLa cells will grow certain spirochetes or not. That's a great question. But what they would do if you were trying to culture these was, instead of using something like HeLa, you'd use some other cell line that was derived from epithelial cells, in the case of syphilis, for instance. But HeLa cells have been used to culture all kinds of different things. So it's a possibility, sure. But you would definitely have to use immortalized cells to do this. And that while we're talking about that, it brings up an important point. A lot of bacteria and viruses can't be grown using normal lab culture techniques. And so until we had genetic sequencing that was so readily available and affordable, uh, there was a lot of microbes that weren't discovered because they couldn't grow under the conditions that we had in the lab. So if we couldn't grow them, we couldn't go through the 6 eyes, right, and identify them. So a lot of microbes that we know about now are just coming to light in the last 20 years with the advent of shotgun sequencing. Okay, the first one I want to get into is treponema, and this is going to include syphilis. Okay, they're going to look like these long, thin, spirally coiled cells. Okay, and again, these require all the things that I mentioned in the previous slide, live cells for cultivation. And this is the causative agent of syphilis. Humans are the natural host for syphilis. So you don't get syphilis from animals, you get it from other humans. It doesn't survive very long outside the host, thankfully. And so they're not typically acquired by like using public restrooms and things. They're acquired by sexual intercourse and through mother to fetus transmission. Okay. There are 3 different stages. There's a primary stage, a secondary stage, and a tertiary stage. And in the primary stage, the syphilis cell will bind to the epithelium, and it's usually in the mouth or in the genital area. There it starts to multiply and form a lesion called a canker at that site where the epithelium was inoculated. Okay. And then this canker will then, of course, lead to fluid secretions that are highly contagious, and that's how it's passed from one person to the next. Over time, these cankers will heal, and before we understood syphilis, it was believed that the infection was over at that point. That's not the case. That's not what is really happening. What's actually happening is that the syphilis is moving from that initial site of infection into the blood system, the circulatory system, to then go on to the next stages of more advanced syphilis. This is going to be the secondary stage. Now you have it essentially systemically in you, and that manifests as a rash on your skin, usually covering your body or in patches. And there could be fever, headache, sore throat that also comes along with that rash. The rash isn't particularly uncomfortable, but it can be persistent. And so oftentimes back a long time ago, people mistook these symptoms, or actually these would be signs, right? These particular signs as 2 different infections. They didn't make the connection that this rash was actually the same thing that was, um, it was the same infection as that initial canker. The rash goes away, and then you get into this tertiary phase, which is a latent phase. It can go for decades without any symptoms, and eventually will lead to things like gummas, which are these painful tumors. Let's see, where's my annotation here? So right here can lead to these really swollen tumor-like masses. You can even, it will infect the nerves that regulate the pupil of your eye. And so it no longer maintains its circular shape. It loses its morphology. It gets irregular. And then you can get these really, really severe, deep ulcers on the epithelial. And again, back in the day, there are these horrific pictures of people missing their complete nose. Like it can eat away your entire nose. It can cause these terrible ulcers all over your body. And by this point, it's pretty much you're past the point of return. And a student emailed me a while ago about syphilis and the role that it played in with Christopher Columbus and stuff like that, and it was pretty interesting how historically, how the intersection of microbiology and history happen, and how things get transmitted, because syphilis was once upon a time, syphilis wasn't in Europe. It wasn't until these expeditions that brought pathogens to the Americas, and then also brought pathogens that were native to the Americas back to Europe. Okay, clear. There's also congenital syphilis, so not acquired through sexual intercourse. It's acquired via the mother who has syphilis, and it can be passed through the placenta. And this can happen at any point during pregnancy. And so oftentimes this leads to really severe abnormalities in the infant, bone deformation, impacts of the nervous system and cognition, and all kinds of deformations skeletally. And there's really this hallmark sign of congenital syphilis is going to be Hutchinson's teeth. So it has these notches in the teeth here, these barrel-shaped erosions, and that is from acquiring it again through in utero. How do we diagnose and test for it? It's challenging because oftentimes the symptoms and signs overlap with other STDs. Just like I was talking about at the beginning, how we have all kinds of illnesses that come around in the fall and winter: rhinovirus, other coronaviruses, influenza viruses that will have the same symptoms and signs as COVID. Same thing with syphilis, in that it's not necessarily distinctive at the initial infection. And so to treat it, or sorry, to diagnose it, you need to take a medical history of the patient, to see what kind of role that might give. Then you'll take samples and you can do macroscopic or microscopic testing. So, you know, take a sample of the canker and look at it under the microscope and see if you see these spirochetes. Or you can do serological testing. So you can do an ELISA with antibodies specific to syphilis and see if you get binding. There are different staining techniques using silver. So again, these spirochetes aren't going to respond to the typical gram stain. So there are other stains that need to be done in lieu of that. And then treatment is usually just penicillin. And if you catch it early on, you know, it's treatment's pretty effective.
What about with like congenital syphilis? Is that the same thing?
Treating,
Yes.
well, I think that by that, you know, depending on when the transmission was, you might be able to treat it. But depending on how long the fetus has been developing with it, it becomes much more difficult because this is not just something that's on the outer surface of the patient, right? With congenital syphilis, it's had time to reach the nervous system. You can't undo any of the skeletal deformation as a result, you know. So I'm not exactly sure what the prognosis is for congenital syphilis. I mean, I know that um, that infants will survive into adulthood usually, um, but oftentimes it's much more difficult to treat um, than if it's contracted via sexual intercourse. Good question. Any other questions on that? Okay. I wanted to move on now into Borrelia, which is an arthropod vector. So syphilis is human to human transmission. Oftentimes, this Borrelia, and actually in basically all cases, it's going to come from a tick or a mosquito or some kind of insect as the vector. And so the one that really stands out is Borrelia burgdorferi, which is the causative agent of Lyme disease. And when you get Lyme disease, it's through a tick, and the tick has maybe acquired it from feeding off of a deer, and then that tick will jump off the deer because they're not really very effective at removing ticks like us. And so now it has that pathogen in its blood. And then it bites a human, and some of the blood in the tick goes into the human and has effectively seeded that human with Lyme disease. And it often will manifest in this bullseye rash, okay, but not 100% of the time. So humans um, can get Lyme disease and not really know it early on. But oftentimes it does lead to other symptoms, other signs, fever, headache, stiff neck, dizziness. And if untreated, can lead to some really bad sequelae like cardiac arrest. What's wrong? You're okay. Neurological deficits, arthritis. It's a pretty bad way to go, honestly, and it's something that some people really never recover, and you know it can lead to oftentimes like chronic fatigue, and some people think that Lyme disease is much more prevalent than what we acknowledge, and that Lyme disease is a causative agent of all kinds of different symptoms that aren't attributed to it. Now, there's really not much science to back that claim up, but it's something that's circulating out there. I believe someone in South Lake Tahoe or in the Tahoe area wrote a book about this topic, and postulating that Lyme disease is much more attributed to certain things than we realize. Now, how do we
I have a
treat?
question.
Yeah.
So if it's proven that microbes are the cause of Lyme disease, then why are so many states or scientists or doctors so Lyme illiterate, I guess? Or like there's a lot of, my friend worked in a Lyme disease clinic, and there's a lot of places or like practices that just don't believe that Lyme disease is real.
Hmm.
I don't know. I'm just wondering why that is, or maybe it's being like proven that it's real and it's become less and less a thing. But
Oh, I mean, it's scientifically sound that this Borrelia is the causative agent of Lyme disease. Now, getting into why some people don't believe that is opening a can of worms that we could talk about the rest of the lecture, right? And that, why do some people choose to reject certain, findings in science? I'm not as aware of scientists and physicians rejecting that Borrelia is the causative agent of Lyme disease, as I am aware of people thinking that Lyme disease is an underlying cause of other symptoms and signs that aren't attributed to that disease. Does that make sense?
yeah,
So I don't know, you could enlighten me, and we could talk about this later on if you want. I wasn't aware that there was a lot of denialism of Borrelia being the cause of Lyme disease, but in this day and age, I don't I don't very little shocks me in terms of what people reject that is scientifically sound. So
it could be other countries. I'm not,
Mhm.
I'd have to talk to my friend again, but I know she had patients from like all over the world that
Mhm.
would come to her office to be treated
Uh-huh.
because there were not places to be treated where they were living. So
And
maybe
that
it's other countries and not
and
in
that's
states.
that's very, I mean, that's very possible. And again, there's nuances between being, between not being able to treat the disease and not thinking that Lyme disease is caused by a microbe and that it's caused by something else, as well as having a more narrow view of what Lyme disease manifests into, right? So they're all, you could, if some people think that Lyme disease isn't caused by a microbe, then the burden is on them to provide something alternative, right?
That makes sense. That sounds a little more correct. Maybe it was had to do with the treatment or the symptoms and
Yeah,
signs.
I mean, I think in terms of what I've come across, there does seem to be people that don't dispute the cause of Lyme disease, but um, do, like I said, think that Lyme disease is the underlying like cause of many other um, uh, health burdens, we'll say, other than just these few symptoms and signs that I've outlined in this slide here.
Makes sense.
Thank you for bringing that up. And guys, always feel free to jump in at any point. I prefer to have a more of a discussion than me just talking. So feel free to jump in whenever.
I have another
Now,
question.
yeah.
How does it spread in ticks? Is it just something that ticks like innately have, or is it like just like there are infected populations in certain pockets? Like, how does that work?
Usually the ticks acquire it from some other animal, and it just circulates from tick to animal, tick to animal, and eventually that tick will then give it to a human. It's just a matter of time before a human gets bit by a tick. Does that make, is that what you're asking?
Yes, that is what I was asking.
Yeah, it doesn't, as far as I know, it doesn't infect the tick. Most arthropod vectors are not infected by the pathogen that they spread. Okay, so a mosquito isn't impacted by malaria in the least. Um, as far as I know, these ticks don't get Lyme disease, right? They just feed on other animals that do have Lyme disease. And this, because Lyme disease won't necessarily kill an animal quickly, they can live long enough to just have it. And so when a tick that doesn't carry Lyme disease feeds on them, then now they have it in their blood and they pass it on to the human that way. Um, I just saw your comment, Sarah, about the link with chronic fatigue syndrome. Yeah, I mean, again, there are things out there. Chronic fatigue syndrome is not well understood. And interestingly, I think I've already said this, so I won't spend a whole lot of time on this. But chronic fatigue syndrome, back, I want to say 10 years ago or so, was linked to a virus. And the researcher who linked it to the virus, this enterovirus, um, turns out that her plasmids that she was using in lab were contaminated. And so they sequenced these samples and they found this virus in the samples, but it was just because it got contaminated in the lab. And then there was some dispute on whether or not they'd actually spiked the samples with that virus. And so that researcher her career was effectively kind of destroyed after that. And more recently this year, she became the person that was interviewed for Plandemic. And I don't know if you guys know about Plandemic. I think it's kind of impossible to see now. YouTube took it off, but it was this completely anti-scientific propaganda put out to link COVID with a conspiracy of the U.S. military and China, and that it was all one big plan. So again, always consider the source of your information. Anyway, yeah, it's I feel like it's highly possible that Lyme disease could be not, if not a contributor to chronic fatigue syndrome, because like I said, that is one of the symptoms. And so I think, Emily, you were the one that asked the question about how it's contracted and stuff. And this is the life cycle of the Lyme disease. So here you can have a mouse that has the pathogen, and then a tick bites it and the mouse, and it's calling it an infected larval tick, but I don't really know if that is if that's accurate or not. I'm not sure if the spirochete actually infects the tick or if it's just a carrier. Then that tick develops into a nymph and can bite a human or can bite a deer, and it just continues to circulate through these animal to tick crossovers. Okay, any other questions on Lyme disease?
How do they test for it?
I believe they usually do a combination of microscopy to look for the actual morphology, and then more commonly they'll do a PCR or an ELISA, some kind of serological test using antibodies that are specific to Borrelia, and then that's how they diagnose it. So, I mean, it's pretty definitive to figure out if you have this pathogen. You just got to go to the doctor and give them a sample of your blood. Okay. Any other questions on that before we move into curviform bacteria? So curviform bacteria are going to be the ones that are, oops, what did I do? Sorry. Not rods, not circles. They're more, kidney bean shape, like that. Okay. And there are three main groups that lead to enteric disease, and that's going to be Vibrio. That's going to be the Vibrio cholera, the stuff that is transmitted in unsanitary conditions where there's a lot of feces around. And so that's going to lead to diarrhea, which then puts more feces into the area. And so it's a kind of a perpetual cycle. There's also Campylobacter. These are going to be spirals, like spirochetes, or curved rods, depending on the species. One flagellum. So what would that be? If you have one flagellum, what are you?
Monotrichous.
Ding, ding, ding. Yes, monotrichous. Great. And then Helicobacter or H. pylori, which is the stomach bacterium that has been associated, well, not associated, is the causative agent of some ulcers, stomach ulcers. It's a spirochete and have several polar flagella. So if they're polar flagella, what do you think that's going to be? Not mono. If it's on either end, it's
Amphi.
yes. Great. Good. Amphi. If there's a bunch of them coming out of one end, what's that?
Lofro. Sorry, I don't want to be the only one answering.
Come on, guys.
Lofro.
Lophotrichus.
Lofo, sorry.
And then if they're all over the cell, it is
Trichus.
what kind of trichus?
Petratrichus or
Oh.
Petri.
Close. Peritrichus, right? Peripheral. Peri is to be all around. So peritrichus. Yeah, I'm asking you all this because none of this will be on the final. So this is your last chance to get this, right? Because the final's not cumulative. All right. Diving in a little bit more into Vibrio cholera, this is going to be oftentimes found in the tropical regions during monsoons, where you have a huge influx of water that floods the sewer systems, or if there isn't a sewer system, just kind of takes wherever that sewage is and homogenizes it throughout the area, and has been a consistent pandemic dating back to the 60s, but it goes beyond that too. It can be spread to non-endemic areas, meaning, so if it's endemic, that means it springs forth from that region. It's localized to that region, but it can spread through the transport of goods, being transport of beverages and food products that are contaminated. And specifically what it infects is the mucus barrier in the small intestine. So that part where all of our healthy gut microbiota live, it will attach there and just really start forcing the water out of your tissues and into your gut, and not your gut, but your digestive tract. And then from there it will lead to just really, really terrible diarrhea. It's called like rice water stool because it's like a cloudy, whitish cloudy, just liquid that comes out. And that is directly linked to the toxin, the exotoxin that the cholera secretes. Now, luckily, it's a non-invasive bacterium, so that means it doesn't penetrate typically into the body. It gives a lot of diarrhea and a lot of discomfort, but it stays in the intestine until it's flushed out. And the reason that it's deadly is not because of the damage it does to the body, but because of the chronic diarrhea, which leads to dehydration. And so really children are the ones that are most susceptible to dying from cholera, because they just dehydrate and they can't overcome that. Okay, so in order to treat it, usually rehydration, lots of electrolytes. Electrolytes have saved many people, because before we knew about electrolytes, it was really hard to rehydrate. Even giving them water, right? It would just pass through them. And so the electrolytes allow the water to actually enter into the cells. And then also, often they'll give tetracycline. Other Vibrio pathogens are going to be found again in these tropical, coastal regions, right, where there's water. And they can come from invertebrates as a vector. And so you can get Vibrio from eating raw seafood. And as a result, that will give you symptoms similar to cholera, diarrhea, vomiting, that kind of a thing. Raw oysters are oftentimes associated with Vibrio vulnificus. And so you can get it from contaminated food and water. You can get it from contaminated invertebrates that are also infected, then you eat them. And so again, the treatment is usually just to replace fluids, sometimes tetracycline. But oftentimes, again, these things, the only reason that they kill is from dehydration. And so if you can hydrate the patient, you can, coax them along long enough to where their immune system fights off the bacterium, and then it's not a problem. Campylobacter. These are going to be these little squiggly guys here, kind of cute. They remind me of nematodes in a way. What kind of flagellar arrangement is on these guys in this picture here?
Bitrachea.
Bitrachea, close. Yeah, amphitrichus, right? Because it's on either end. So let's see here, right? So we got a cute little flagella right here and a cute little flagella right there. So one on either end. That's going to be amphitrichus. Now, if we had a bunch coming off here, that would be lophotrichus, right? Okay. These are going to be in the intestine, genitourinary tract, but also found in birds and mammals. And so humans that eat different birds and mammals can contract it if they don't eat properly cooked birds and mammals. And oftentimes is of medical relevance in veterinarian situations. So pets and livestock can get Campylobacter jejuni and Campylobacter fetus, which can lead to the demise of these animals, and so is a burden economically and financially for farmers and things. So jejuni is going to be in humans transmitted by beverages and undercooked food. It's going to also infect the mucosa of the small intestine. But unlike uh, unlike cholera, it's going to burrow into that mucus, okay, and get closer to the epithelial cells. But again, it like cholera, it can excrete this exotoxin, or in this case, they call it enterotoxin because it's a gut microbe that leads to diarrhea. And so that diarrhea can come also with headache, fever, bad abdominal pain, and then bloody or watery diarrhea. And it's bloody, unlike cholera usually isn't bloody, but it's bloody with jejuni because, again, it's burrowing into the mucosal layer. So it's getting beyond that and perturbing the intestinal epithelium. So like with cholera, you're going to rehydrate. You might use some antibiotics, in this case, erythromycin or azithromycin if it's a severe case. But, in general, the way to prevent this is to pasteurize milk, cook your food properly, prepare your food before it's cooked properly. And oftentimes that is sufficient.
I have a question.
Yeah.
Since it burrows in the mucus layer and it gets closer to the epithelial layer, does it cause inflammatory responses at all?
Sure, absolutely. So anytime you, that's a good question. Anytime you get to that epithelium, the receptors on the epithelial cells are going to recognize the foreign invader and then start secreting signaling molecules that will then bring in the inflammatory response. And so, yeah, absolutely. Good question. Okay, Campylobacter fetus is going to be, again, oftentimes linked to livestock. And so if the livestock happens to be pregnant, which is where it gets the fetus species name, it leads to an automatic abortion of the fetus. It can't survive the infection. And so again, as you can consider, if the livestock can't reproduce efficiently, that's going to put a burden on agriculture. Now, interestingly, it also gets, I don't know if I think it, I don't think it gets the name fetus from this, but coincidentally, I guess it can be opportunistic in women that are in their later stages of pregnancy. So the third trimester of pregnancy, pregnant women have all kinds of hormonal and immune modulation going on as a result of having their own little darling parasite inside of them. And so it makes them more susceptible to certain diseases. And this is one that they can get opportunistically. And as a result, if it, it may, while it kills livestock, if the human newborns survive, it's going to lead to things like meningitis and pneumonia. So much worse than typical Campylobacter infection in humans that just is a digestive or an entero disease. Okay. And that brings us to H. pylori. Again, it's got these curved spirochete type cells, and is the primary agent in the development of stomach ulcers. Okay. And as a result, can also lead to stomach cancer. Okay. So when you have these ulcers form, it can lead to mutations in the epithelial lining that are trying to heal that ulcer, and then can lead to different stomach cancers. So prolonged stomach ulcers can be later on driving cancer. And interestingly, people with O blood type have a higher rate of these H. pylori induced ulcers, which I think is interesting, because there's just a study that came out recently showing that O blood type humans are less likely to have severe, well, no, wait, let me get this right. I think they were less likely to get COVID, I want to say. They weren't able to determine whether or not the symptoms were more or less severe, but when they did this analysis, this meta-analysis of COVID patients in the hospital, and they normalized for the different representation of the blood types, they found that O blood type was less susceptible, which is interesting. I don't really know the mechanism behind how blood type can confer susceptibility to pathogenesis, but there's definitely some link there. Okay, any questions on those three major forms, the cholera, the Vibrio, the Campylobacter, and the H. pylori? Because if not, we'll move into rickettsia. So this is going to be an intracellular pathogen that is passed on through an arthropod vector like Borrelia. And so we'll talk about rickettsia in a moment. We'll also talk about a little bit about chlamydia, which is also an intracellular pathogen, meaning these bugs need cells to survive. They don't really survive very long outside of a host. And then, and also worth mentioning, they go into the cell of the host, right? Not all bacterial infections internalize into the cell, right? And then mycoplasmas, those are the pleomorphic cells that are made up of sterols and have kind of an amorphous shape to them. So rickettsia is gram negative. It's non-motile, so it doesn't have any flagella. And it's an obligate intracellular parasite. So it has to be inside a cell in order to reproduce. It can be transmitted through ticks and fleas and lice. And that's how humans get it. They get it from being bitten by one of these arthropods that has it in its blood. And from there, the rickettsia will enter into the endothelial cells, leading to a breakdown of the vascular lining. Okay. And now you have things like, vascular leakage, or like, serum and blood coming out, and things like thrombosis. Okay. So, pretty severe disease. And this is a picture of right here. This is a bacterial cell on the outside, and then below we're looking at a bunch of bacterial cells intracellularly. So they're inside the host cell now, and they're multiplying. Two main types of disease that are caused by rickettsia that's called rickettsiosis. So there's the typhus, epidemic typhus, and endemic typhus. These are going to be passed on via lice for the epidemic typhus, leading to fever, chills, headache, rash. Again, very common symptoms in microbial pathogenesis. So it's hard to determine right off the bat what's causing those signs and symptoms. And then endemic typhus is going to be usually through fleas. So you live in an environment that has an infestation of mice or rats or both, and they get bitten by fleas, and they give that rickettsia to the flea, and then the flea bites humans, passes it on. Luckily, endemic typhus is not as severe as epidemic typhus, and I believe that's where they get their names. Endemic is more localized, epidemic is more widespread, and then of course pandemic is worldwide. And then the other big one is Rocky Mountain spotted fever. Okay, this is going to be, again, an arthropod vector, and it can come from your dog. So, your dog gets bitten by a tick, gives it to the dog, and then the dog gets bitten by another tick, and that tick, comes off it and bites you. Or it skips the dog altogether. You don't have to have a dog to get Rocky Mountain spotted fever. You just need to have the ticks. Okay. It does have a distinctive spot-like rash, and if gone untreated, can lead to severe heart damage and damage of the central nervous system. Okay. So this is a disease that's present in the US. And again, is something to be taken very seriously. It doesn't contribute to a high burden overall in the US population, but those that do get it can have very severe outcomes. And so that makes it a worthwhile pathogen to study. Okay, here's just a table looking at rickettsiosis and the vector that causes them, the primary reservoir where they are existing and being passed from, modes of transmission, and where they're located geographically. In the US, as you can see, the highest incidence of Rocky Mountain spotted fever is going to be in the like middle, Midwest, northern, southern regions. Okay, so Missouri, Arkansas, those places are going to have higher incidence of Rocky Mountain spotted fever, which makes sense because the Rocky Mountains are around this area. So that's that's the Appalachian. The Rocky Mountains are over here. So I guess let's see where there are some, yeah. So in yellow, that's second highest level. There are some significant incidents in the Rocky Mountains as well, and that's probably, I guess that's where it got its name, because maybe that's where it was first detected. But maybe someone can enlighten me. I know someone's doing a Rocky Mountain spotted fever proposal. Do you know how it got its name by chance?
I don't know if I recall exactly. I know that they were studying in that area because it started in Idaho, but it wasn't called that yet. And then they developed the Rocky Mountain spot laboratory, and then that's where they did all the testing and stuff. So maybe
Interesting. Okay. Yeah, that makes sense.
but
Yeah. Okay. Rocky Mountain was the lab, and spotted fever because it causes a spotted rash, and one of the signs is a fever. So, okay, cool. A lot of these pathogens have interesting names, whether it's their scientific name or the disease that they cause. They can be quite creative and colorful at times, and always have some kind of story to them. So this is the rash. This is that spotted rash. You know, kind of looks like the secondary phase of syphilis in a way, right? So again, oftentimes these pathogens are hard to diagnose at first without some kind of molecular analysis to go with it, because just observational diagnoses isn't enough to narrow it down. And so it has a 20% fatality rate, and 20% is high. I don't, maybe it doesn't seem high, but if you think about it, 20% is quite high, right? What's that? One in 5 people that get Rocky Mountain spotted fever are going to die from it. So not something to be taken lightly. So Bartonella, also related to rickettsia, and small gram negative, and it's one of the emerging pathogens. So we talked, remember, about emerging pathogens and re-emerging pathogens and the difference between those. So this is one that is newish, right? And is associated with trench fever from lice, and something we've probably all heard about, cat scratch fever. And so that's going to be something that is also more severe in AIDS patients because they're immunocompromised. Okay, so they cannot fight off this infection as well as someone who is immunocompetent. Chlamydia, also gram negative, like some of these others, is obligate intracellular parasite. Okay, so it has to go inside of a cell to replicate. And this is a diagram of the life cycle. There are 2 different stages. There's an elementary body where it's essentially metabolically inactive, kind of like an endospore in a way, but not an endospore. And then the reticulate body, which is non-infectious, but that's what's actively dividing and growing in the host. Okay, and it forms these, it divides within these vacuoles, or these compartments inside the host cell, right here, RB, so reticulate body. And then they're released, and then they go into the next cell. And this cell here is a phagosome, so they infect immune cells, right? Potentially. You get chlamydia from other humans, okay? There are 2 different types. There's trachoma, which is going to attack your mucous membrane in the eye, in the genitourinary tract, and even your lungs. And then there's the lymphogranuloma, which invades lymphatic tissue present in the genitalia. Okay, so it's a sexually transmitted disease, and can't blame the arthropods on this one. We can only blame ourselves. Ocular trachoma, so that's going to be, again, this first one here, this trachoma, and this is an example of that. So they've lifted up the eyelid, and you can see underneath the eyelid there are these like pustules here, and even on the eye itself, it can destroy the cornea. And so once you destroy the cornea, you're blind, right? You can get it also if the mother has it. It can be passed on through the birth canal, and what leads to inclusion conjunctivitis. So also in the eye. Now, if it's properly screened ahead of time, then the mother can be given certain prophylactic antibiotics to um, to treat or dampen the chlamydial outbreak, and so the baby, can be born free of this conjunctivitis. Nasty stuff. Okay, and then that brings us to the last one, the mycoplasmas. Okay, and these don't have cell walls. They have these sterile components of their membrane, and so they don't have the structure and the rigidity of other bacterial cells, and therefore they're going to be pleomorphic. They aren't going to have a particular shape. They're not rods, they're not coccus, they're not spirochetes. They are unto their own particular shape. Some of them cause pneumonia, and so is going to infect the lungs, and then from there, moving on to other areas, leading to fever, which is, again, fever is one of those first responses that is generalized to all different types of pathogens. But then also chest pain, sore throat can accommodate, or not accommodate, but can come along with fever. There's Mycoplasma genitalium and uroplasma, and so those are going to be the STDs, as the name implies. And so there are multiple ways to get these Mycoplasma bacterial infections, and they can infect, depending on the species, different tissues in the human body. Okay, I think that's it. We're all done. Any questions about that? Okay, these are the unusual suspects of medically relevant bacteria. And so next week we're going to talk about the more usual suspects of medically relevant bacteria. Okay, and so hopefully this is exciting, because now we're really drilling down into the stuff that impacts human health and disease, and the stuff that, for a lot of you going into the medical field will, this will be relevant, hopefully, to your job someday. Not hopefully, because hopefully people get it, but like, hopefully this stuff will be important to know. So with that, have a great weekend. Be on the lookout for a message from me setting up a time to meet to talk about proposals for Monday's lab period. If you don't get a message from me, again, you've got lab off. Do it with it as you wish. Hopefully you still work on your proposal, though, because it's due next week. Fungi kits will be ready to pick up Wednesday, and Friday we will really try hard to do our virtual online lab. Any questions before we sign out? Or
Yeah.
comments? Yeah.
If we don't get a message from you, is it going to be about if we just like me and my partner, we're kind of hoping that you would want to look over our rough draft again on Monday, like after we work on it this weekend?
Yeah.
Maybe
So
do that.
hopefully you've all looked at the comments and feedback I gave you. If not, it will be there. And if you would still like, if you don't get a message from me, and Emily, you're assuming that you won't get a message from me. If you don't, I'm still happy to talk about your proposals. It doesn't mean that that I'm not here for you guys to bounce ideas or give you feedback on. Okay, so it just means that there are some other proposals that I'd really like to prioritize in terms of from the start, because it's due next Friday. And if we don't get to if we don't have time for a one-on-one Zoom Monday, we can always schedule it some other time.
Okay.
So I think my wife is off on Monday, so uh, I should be unencumbered by littles, and so we can let me see how much time it takes for me to talk with people, and if there's time left over, we can schedule something.
Okay.
Okay. Okay. Well, you guys are free to go. If you do have any other questions you'd like to bring up or comments at this time, feel free. Otherwise, I will see you on Monday for lecture or for our little chat.
Professor, I was actually just trying to mention earlier if hoping you could probably post the slides prior to lecture, just so I could. It's a little hard to keep up with the lecture and notes.
Sure. I
Okay.
did. The slides should be posted.
Oh, okay, great. Thank you.
Yeah, I always post the slides before lecture. Sometimes not much before lecture, but they're always there before we start, so that you can look at them. And I know that sometimes I go kind of fast through this stuff. So always check the week pages in Canvas. So right now I think we're on week 9. So if you go to week 9, the slide decks will be there.
Oh, okay. They're probably just not loading on my end. I'll double check. Thank you.
Okay, yeah, you're welcome. All right. Theo.